Welcome to the Week 6 overview. This week we'll be discussing the manufacturing of biologics. Our primary examples will be biosimilars, enzymes, and antibodies. The market for biologics is a rapidly growing one. The market for enzymes, for example, is a $5 billion industry, and it is growing at an annual rate of 6%. The antibody production market is a $2 billion industry, and it is growing at a 12% annual rate. Within the enzyme market, many enzymes are produced for biotechnology applications within the food manufacturing industry. Enzymes are often produced in bulk, where the emphasis is on high efficiency and low cost. Within the antibody market, antibodies are generally high-value products, and the primary concerns are purity, stability, and specificity. There are two primary statutes which regulate the manufacturing of biologics. First, the Public Health Service Act. This contains the Biologics Price Competition and Innovation Act, or BPCI. This was passed as part of the Affordable Care Act in 2010. The BPCI is an abbreviated licensing pathway for biologic products shown to be biosimilar to or interchangeable with an FDA licensed reference product. Second, the Federal Food, Drug, and Cosmetic Act also contains an abbreviated new drug application process in Section 505, which was also established through the 1984 Hatch-Waxman Amendments. It was done generating the generic drug program for small molecule drugs. The term biological product has now been defined to include proteins. Under the BPCI Act, a protein, except those that are chemically synthesized, are regulated as biologic products. Historically, some proteins have been approved as drugs under Section 505 of the FDC Act, and other proteins have been licensed as biologics under Section 351 of the Public Health Service Act. As we move forward, it's important to remember that biologics are much more complex than the small molecule drugs, both in their structure and in their biomanufacturing. So biologics are drugs that are made from and or are expressed by living systems, and this can include microorganisms, plants, animals, and even humans. Biologics work in concert with the body to address an underlying disease state, not just the symptoms. For example, a biologic can upregulate desired effects, such as erythropoietin for the treatment of anemia. They can downregulate non-desired effects, such as Remicade for the treatment of rheumatoid arthritis. Or they can augment a body's natural defense mechanism, such as Factor VIII for the treatment of clotting disorders. So remember, biologics are made by living systems. They tend to be less toxic, and they have a tremendous potential to treat unmet medical needs. So three examples of the topics that we'll cover are biosimilars, enzymes, and antibodies. So what is a biosimilar? A biosimilar is made by copying an approved biologic drug. The same concept as a generic drug copies an approved small molecule drug. First, you select an approved drug, you replicate its chemistry, manufacturing, and controls. You wait until the patent expires or invalidate the patent. You demonstrate the equivalency, and you register it for sale in the market as generally cheaper and or faster than the original drug. Now there is a major caveat. Copying a biologic is far more difficult because it's not always possible to precisely replicate the original environment and or living system that was originally used. So when we talk about replicating the chemistry, manufacturing, and controls, What does that mean, and how does that affect the drug we're selling? Well, the Chemistry, Manufacturing, or CMC assures that a drug sold to the public has the quality attributes similar to those of the original drug in terms of safety and efficacy. The CMC assures that the quality of the drug meets the appropriate standards and is consistent. The CMC assures that the drug that you are using is the drug described on the label. And the critical elements of the CMC include how and where is the drug made, how are the raw materials tested and monitored, what control procedures are used to assure product consistency and quality, are quality attributes adequately identified and characterized, are the test methods used to monitor the product qualities appropriate, and how long does the product maintain its quality after it is made. In many instances, a biosimilar is compared or likened to a generic drug. So how do they compare? Well, for generics, these are small molecule drugs that are synthesized in facilities by chemical reactions to create a chemical entity. The same chemical entity can be made in a different facility by a different company, same synthetic method. In contrast, biosimilars, because biosimilars are made from living hosts, one company's drugs will be inherently different from another's. Small transient changes in the host cell or during manufacturing can cause changes in the final product, such as post-translational modifications, such as glycosylation patterns often differ, or proteins may fold differently. So why is there so much interest in biosimilar drugs? Currently, drugs or pharmaceuticals accounting for more than $20 billion in annual sales have lost patent protection. The global biosimilars market has been valued at $1.8 billion in 2013 and approximately $2 billion in 2014. This market is expected to reach $4 billion by 2019, with a compound annual growth rate of 15% between 2014 and 2019. It's important to also note that biologics are a growing segment of the drug population, with 4 of the top 5 oncology drugs being biologicals. What are the regulatory issues associated with biosimilars? In 2012, the FDA issued the draft guidance document on biosimilars. This set key requirements for biosimilarities. One of the primary issues is the primary sequence of a protein, the amino acid sequence, must be identical. It also introduced the concept of totality of the evidence approach, which is used by the FDA with comprehensive analytical analyses, animal and clinical data to assess biosimilarity. The FDA's draft guidance document outlines the totality of the evidence approach. This document describes the stepwise approach to evidence development. It ensures that development includes only those elements necessary to address residual uncertainty. It introduces the concept that only after a thorough review of data from structural and functional analyses can the FDA provide meaningful advice on the scope and extent of necessary animal and human testing. It explains the general expectations for human clinical trials. Within these trials, at least one study is expected to present the immunogenicity or the pharmacokinetics or the pharmacodynamics. Comparative safety and effectiveness data may be necessary if residual uncertainty. Within the BPCI document, a 351 application must include information demonstrating that the biologic product is 1. Similar to the reference product, 2. Utilizes the same mechanisms of action for the proposed condition of use, to the extent known for the reference product. 3. The condition of use proposed for labeling have been previously approved for the reference product. And 4. The product has the same route of administration, dosage form, and strength as the reference product. For the general requirements of a 351(k) application, this application must include information demonstrating biosimilarity based on data derived from 1. Analytical studies which demonstrate the biologic product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. 2. Animal studies including the assessment of toxicity. And 3. A clinical study or studies including the assessment of immunogenicity and pharmacokinetics or pharmacodynamics that are sufficient to demonstrate safety, purity, and potency in one or more appropriate conditions of use for which the reference product is licensed. What are the steps involved in the analytical analyses of biosimilars? First, there is a side-by-side comparison to the reference product, and each assay is required to show comparability. Structural assays to assess structure and secondary modifications are required. And functional assays to assess target binding, cellular effects, selectivity, and specificity are also required. So in summary, biosimilars. Biologics or biotherapeutic drugs are made from living systems, hence they are large and complex and are impossible to make generic. They are naturally occurring, less toxic, and work with the body to serve unmet medical needs, for examples in the fields of oncology, autoimmunity. So what are some of the key opportunities? Biologics are much desired and beneficial to the global patient population. It is a large and profitable and growing market. There is increasing worldwide acceptance of biologics. There is less competition than in the small molecule drugs, coupled with the potential for the niche markets. Interchangeability will greatly increase the opportunities for biosimilars. So what are some of the key challenges? Biosimilars are more expensive and/or more difficult to manufacture, analyze, and possibly more difficult to prove equivalent. Regulations developed in the U.S. may not be clear elsewhere. Long market exclusivity for reference products may prevent the development of enzymes. What are enzymes? Enzymes are proteins with their associated tertiary and quaternary structures. They act as catalysts to accelerate a reaction. They do not permanently change the process. And enzymes work by weakening bonds that lower the energy of activation. This slide shows a scheme for the production or biomanufacturing of an enzyme. So if a model system produces a particular enzyme following genetic engineering, the enzyme can either be extracellular, i.e. secreted, or intracellular. If it's intracellular, it requires the rupture of those cells. And as we talked about in the downstream or upstream processing areas, first you have to remove the cells by filtering and then concentrate, precipitate, and chromatography as approaches to isolate impurities. Antibodies. What is an antibody? An antibody is a protein used by the immune system to identify and neutralize foreign objects, such as bacteria and viruses. Each antibody recognizes a specific antigenic epitope unique to its target. Polyclonal antibodies are antibodies that are derived from different cell lines or cell populations. Monoclonal antibodies are antibodies that are identical since they are derived from a single parent B cell. Now, there are multiple different isotypes of antibodies, and these are based on their heavy chain constant domain, and they're grouped into 5 groups or isotypes. There's IgG, IgA, IgM, IgD, and IgE. And these can be subdivided. For example, IgG is divided into IgG1, IgG2, IgG3, and IgG4. In most cases, when one is talking about monoclonal antibodies, one is generally talking about an IgG molecule, which has this classic Y-shaped structure where the antigen binding site is located on the amino acid terminuses of the variable and constant regions of the antibody. What are some of the safety issues concerning the use of monoclonal antibodies as a biologic? Well, first, there's the issue of immunogenicity, or the ability to generate an immune response within the patient. Since the monoclonal antibodies are generated in mice, one of the problems is the production of human anti-mouse antibodies, or HAMA, H-A-M-A. This results in a loss of efficacy by binding up the antibody product and increasing its clearance. There can also be an adverse effect, such as anaphylaxis, which is an immune cross-reactivity which binds the patient's own antibody proteins. As a result, biosimilars must have a comparable molecular profile regarding potential immunogenic areas. You must understand it cannot have an altered glycosylation, an altered aggregation properties, and you have to study any changes that may lead to immunogenicity upon storage of your biologic product. How do we circumvent this immune response to the monoclonal antibodies? Mouse monoclonal antibodies with high affinities and specificities are generated using conventional hybridoma technology. However, the clinical efficacy can be compromised by the generation of human anti-mouse antibodies, which can lead to an allergic or immune complex hypersensitivity. As a result, chimeric monoclonal antibodies have been developed. These chimers combine the human constant region with the intact mouse variable region, and that way the affinity and specificity remain unchanged. However, these can still cause a human anti-chimeric antibody response. The more recent development has been humanized monoclonal antibodies. These contain only the complementary determined regions, the CDRs, or the antigen binding site of the mouse variable region, grafted onto the. So how can the production of monoclonal antibodies be transferred to biomanufacturing? Well, currently there are 3 primary pathways. There are phage display libraries, transgenic plants, and transgenic animals. So what are some of the considerations for biomanufacturing, or some of the issues and limitations? Well, with most cases in biomanufacturing, the first step is the selection of the appropriate cell model for production. And this must take into consideration the importance of post-translational modifications for biologic activity, including such things as protein processing, phosphorylation, and glycosylation. Showing the importance of the appropriate cell line selection, here we show 2 cell lines, A and B, taken through various processes where you can have the generation of antibodies from anywhere in cell line A from 139 milligrams per liter to cell line B where you can have 2,800 milligrams per liter produced. Why is glycosylation important? Many complex proteins, including antibodies and enzymes, are glycoproteins. And these can contain 2 to 30% carbohydrates. Glycosylation can affect a protein's half-life, that is, its pharmacokinetics, and its immunogenicity in the patient, as well as the binding affinity, activity, and stability. Glycosylation is complex. Carbohydrates can be attached to the protein either via asparagine, which are N-linked, or serine-threonine, which are termed O-linked. To address this, there have been a number of modifications on how to glycosylate some of these proteins. One can view sugars on proteins are like accents on the spoken words. So the goals in using biomanufacturing in such microorganisms as yeast is to producing humanized glycosylated therapeutic proteins in a large volume that has certain cost advantages.